Hi, my name is Rachel Thomas, and I'm going to be talking about how a powerful family of AI algorithms, known as deep learning, are being applied to high impact and important problems in immunology. I've spent the last 20 years focused on mathematics, data science, and machine learning. In particular, for the last 12 years, I've been focused on deep learning. Why deep learning? It's a powerful method for finding patterns in data. And the reason we have seen so many breakthroughs in this field in the last decade are due to a combination of the availability of much larger data sets, faster computers that were developed by the video gaming industry, and novel algorithms that have led to significant breakthroughs. Most headlines you hear about AI or new products featuring AI are using some form of deep learning. And I'll explain more about what deep learning is as we get into this. However, a few years ago, I decided to go back to school for immunology. Why did I decide to make this big pivot? Well, the science journalist Ed Yong has warned that we are now living in a pandemicene, a period with increasingly likely pandemics. We're seeing an increase in the spread of infectious diseases for a variety of reasons. Climate change is crowding species into new habitats and into closer and closer contact with humans. Also, frequent global travel in megacities allow unprecedented opportunities for viruses to mutate and for infections to spread from person to person, crisscrossing the globe. Antibiotic resistance has risen dramatically. And at the same time as all this, we're learning more and more about the long-term consequences of infections and how they can contribute to autoimmune diseases, neurodegenerative diseases, and cancer. The human immune system is incredibly complex, and much is still unknown about it. Immunology, virology, and microbiology will become even more important in the coming decades. I decided that these problems are so important that I wanted to be fully informed about them and able to focus on them. Today, I'm going to start with sharing kind of a brief, incomplete history of deep learning and machine learning and explain what those things are, how they're being applied to three key problems in immunology, as well as what the risks, gaps, and opportunities of doing so are. There's no way that I could completely cover this history in just a few minutes, so I'm going to focus on a few key moments in the history of deep learning. So the term machine learning was coined in 1959 by IBM researcher Arthur Samuel. And Arthur Samuel, a non-machine learning research way of doing things would be to program a computer to play checkers with handcrafted relevant rules, such as the rules of checkers. However, machine learning is about learning from previous data. And Arthur Samuel wanted to create a computer program that could learn to play checkers better than the person that had created it, him. He wrote that rather than specifying methods in minute and exact detail, that computers could instead learn from data. And this is the core idea of machine learning. To take a different example, if you're interested in protein folding, a rules-based non-machine learning approach would be to have a score about hydrogen bonds, whereas a machine learning-based approach would be focused on learning from previous protein structures. Machine learning is all about algorithms learning to match inputs to outputs. And so it really depends on what are the inputs and outputs. What is the shape and format of the input and of the output? And so these are helpful questions to ask when you are reading about a new use of AI or machine learning, to think about specifically what these are and to think about what data it was trained on. Because the data that was used as training input will really determine kind of how accurate and what kind of performance this algorithm can have. So moving ahead to the 1990s, the US Postal Service wanted a computer program that could automatically scan letters and convert handwritten zip codes into what the kind of numerical zip code is to automatically sort the letters. People's handwriting varies so much. There are so many different ways to write the digits between 0 to 9. Some of these ways can be quite sloppy. And so the challenge was to come up with a program that can do this. This is an example of a handwritten 8. It's kind of crooked, but you can see here that it can be represented just by a bunch of numbers, the values of how dark each pixel is in the 8. A team of researchers led by Professor Jan LeCun came up with a solution for this that was implemented by the US Postal Service. And this solution used neural networks. So neural networks are a family of algorithms that involve a lot of matrix math. If you took math in 10th or 11th grade, you may have learned to multiply matrices, and neural networks involve kind of scaling that up, a lot of matrix multiplications mixed in with other math operations. Deep learning refers to multi-layered neural networks. It's a specific type of machine learning, and for the rest of this talk, I will be focused on deep learning in particular. And there are many different types of neural nets within deep learning. So it's kind of a family of algorithms. Another key moment in the history of deep learning comes from the 2012 ImageNet competition. So ImageNet is a competition that asks, given an image, what is it a picture of? And so, for example, you might have a picture of a container ship, and teams will enter computer programs that have to determine what this is. And one of them might say, well, this is very likely to be a container ship, and maybe there's a small chance that it's a lifeboat, and then a tiny chance that it's an amphibian or a fireboat. ImageNet has it has 1,000 categories and involves over 1 million training images. And while ImageNet started in the 2000s, it's the 2012 competition that really made headlines. The winner that year was an algorithm called AlexNet. And AlexNet had a number of innovations that allowed it to effectively use deep learning to have performance that was far superior to all the other competitors. And so 2012, AlexNet is really what helped popularize deep learning and break it into the mainstream, even though, as I mentioned, people were developing deep learning algorithms prior to this. And then the final moment I'll talk about in our abbreviated history is the 2020 CASP competition. So this is a different important competition. This is the Critical Assessment of Techniques for Protein Structure Prediction. But basically it's a competition where, given an input sequence of amino acids, computer programs need to predict what the 3D structure of that protein will look like. Amino acids are the building blocks of proteins. And this competition started in the 90s. It's held every other year. And the winner in 2020 was AlphaFold. AlphaFold really smashed the competition. So here, this is a chart showing a measure of the error. So lower scores are better. 146 teams entered that year. This is just the top 10 teams. AlphaFold's here in blue on the far left, and you can see its error is significantly lower than even the second place team. So again, this is just the top 10. The other 136 teams had even higher levels of error. And this is a pattern that is frequently seen when deep learning is introduced to a new field, where it can significantly improve what the state of the art is. Here are 3 particular proteins that were predicted by AlphaFold. So blue is, or rather I should say, blue is the AlphaFold prediction, and green is the true structure that had been determined experimentally. In particular, notice that this protein, labeled D, has over 2,000 amino acids in it. There were no structural homologs already in Protein Data Bank for it, and yet AlphaFold came up with a pretty accurate prediction for it. I'm going to share just one of the key ideas that was used in AlphaFold. And so backing up a bit, there was a very influential paper in 2017 called Attention is All You Need about language models. And so deep learning language models are used for things like translating between languages if you want to translate from English to Turkish. And they're also used by chatbots, so generating new text of what may come next after a particular prompt. So the idea with attention is that to understand a particular word, so here we've got a sentence, the animal didn't cross the street because it was too tired. If you want the model to gain information about it, it needs to know what other words in the sequence to focus on, to pay the most attention to. And attention can let it learn that here animal is the most significant other word in the sequence for determining kind of meaning or significance of it. Attention is now very widely used in language models. And one of the innovations of AlphaFold was expanding this to two-dimensional attention. So in a sequence of amino acids, knowing which amino acid ahead or before the amino acid you're focusing on is most important, which amino acids to pay attention to matters. However, you also want to look at similar sequences of amino acids from other species and know which amino acids in those similar sequences to pay attention to. These are called multiple sequence alignments, and they're a very useful input to protein folding models. And so one innovation from AlphaFold was to have a method for two-dimensional attention that let the model learn both what amino acids in the same sequence are most significant for understanding a particular amino acid, as well as what amino acids in other related sequences are significant to focus on. The reason that I've talked about AlphaFold a bit is that it is being applied to other problems as well that are related to protein folding, such as drug discovery and T-cell binding. In other areas, it's not always clear if AlphaFold is the best approach or not, but it's helpful to know about because even when people are using different types of deep learning, they often will compare or mention AlphaFold to try to offer a comparison. And so with that, I'm ready to dive into the three applications of deep learning applied to immunology that I want to discuss today. The first is a question of, given a protein and a peptide, and a peptide is actually just kind of a short protein, will they bind to each other? And this is a really important question because it's significant for knowing what our T cells will respond to. T cells are a crucial part of the immune system. T cells can directly kill cells that have been infected by a virus or have turned cancerous. T cells also mobilize the response of other immune cells by activating them and sending them helpful information to coordinate the immune response. However, T cells can only recognize peptides when they're presented on MHC molecules. So there's kind of just a very specific stimulus that T cells are able to respond to. And this raises the questions of which peptides will bind to MHC, because that determines what can your T cells respond to, which is really asking what will the adaptive immune response coordinate against. This has direct therapeutic applications. For instance, it's used in cancer vaccines. Cancer vaccines are one type of treatment for patients with cancer. The way they work is that tumor tissue is removed from a patient with cancer and sequenced to identify mutations. And an algorithm is then used to identify which mutations would be most likely to inspire a T cell response. And so to figure this out, you need to know which mutations are things that could bind to MHC and then have a T cell bind and coordinate the response, because those will be injected back into the patient as part of a vaccine. They're paired with a danger signal called an adjuvant, and when they're injected back in, that gives a message to the immune system of, hey, here is this protein we want you to attack that's paired with this danger signal telling you to attack. Deep learning has been used for this in clinical trials, including the pipeline for the PGV-001 neoantigen vaccine trial, but it has been used in other clinical settings as well. So this is a very direct application. There are many, many different deep learning models that have been developed for this problem. I'm just going to share about one of them today. This is an interesting network where The authors took AlphaFold, and here AlphaFold is represented with these red circles, this structure, and it's predicting 3D protein structures. So shown here is these blue spirals. And these researchers took AlphaFold and then stacked another model on top of it, which is just a binary classifier to predict, yes or no, will a particular protein and peptide bind. Then they trained this whole thing. So they were updating the parameter weights within AlphaFold as they trained to customize it for this particular problem of classifying something as binding or not binding. And they achieved great results with this. So these are the ROC curves for both class 1 MHC molecules and class 2 MHC, and the difference between this is just whether it presents peptides to helper T cells or cytotoxic T cells. In both graphs, green shows the performance of AlphaFold. Purple is the combined prediction classification model in the paper from the previous slide. And then pink is the state of the art. And so the state of the art here is a closed source model called NetMHC-Pan. Because it's closed source, we don't know exactly what algorithm is used or what the training data is. But the purple, the model from this paper, came very close to matching it, and it did significantly better than than AlphaFold that hadn't been customized for this problem. The paper also notes that the model shown in purple, it generalizes better to some related problems around protein binding, and it also was more flexible on longer peptides than NetMHC-Pan was. So it was a kind of general and more flexible model. This is just one of many different models that have been developed for T-cell MHC peptide binding. So everything listed in this table uses deep learning. I have divided them up based on which ones are based on AlphaFold or the related models, ESMFold and RosettaFold, which are all using somewhat similar ideas, versus other deep learning approaches such as convolutional neural networks or ResNets. So again, these are all deep learning, but kind of two different broad families of it. You're probably wondering, well, which one of these is best? And that is really hard to determine. Many of these are dealing with different variations of this problem. So some are just looking at peptide binding to MHC, where some look at the peptide MHC complex binding to the T cell receptor. To give one example, MHC fold is more accurate on the TCR alpha chain, but less accurate on the peptide. And so it's hard to come up with, who's the winner here. Also notice all these papers are from within the last 4 years. They're all relatively recent. This is an area that is not very standardized, and some researchers have called for more standardization for a grand challenge in this area that can have kind of more standardized metrics and data sets that'll make it easier to compare which methods really are better than others. But this is part of why I think this area is so interesting right now, is because it is so unclear and there are so many different approaches being taken. All right, the second problem in immunology that I want to tackle is which immune cells interact with which cytokines. So cytokines are chemical messengers that help the immune system coordinate. In this graph, the cytokines are the little white labels written on the arrows. And so for here, you can see we've got 4 different cytokines: IL-10, IL-13, IL-25, IL-27, and they have terrible names, all inhibiting this particular message from this cell to this one. So there are a lot of different relationships. This is actually just a zoomed in version of a even larger graph. And cytokines are really crucial for the proper functioning of the immune system. They're necessary for us to coordinate immune responses. They send very helpful information between cells, but they can go awry in some diseases and cause harm in that case. You may have heard about cytokine storms in the early days of the COVID pandemic, in which an out-of-control immune response to COVID could cause harm and even lead to death. You also see maladaptive cytokine responses in many cancers or autoimmune diseases. And so for this reason, there are drugs that target these maladaptive responses, both existing drugs and future drugs under development, cytokine-based therapies or cytokine inhibitors. And so that's why it's really crucial to understand cytokines and which cells they are conveying messages between. So this is a diagram of a tumor microenvironment. And here the cytokines are labeled on the green ovals. I mean, you'll see this green square is a list of 5 different cytokines, but there are a lot of cytokines involved in these this is a maladaptive response to cancer. So this is one of the reasons why we would care to understand these relationships. So there's a paper tackling this problem that I really like because it uses an important technique known as transfer learning. Transfer learning is based on the idea that neural networks learn information that will transfer to new problems. And so here, the authors took work from other researchers that had created a model called SciBERT. And SciBERT was trained on over 1 million scientific papers. And for this particular study, researchers fine-tuned SciBERT to 60,000 immunology papers. And so that's kind of like they customized it, retrained it to update the weights, but starting with those weights from SciBERT. And this is a super useful technique because it allows people to train faster and with less data. This is part of what has made deep learning kind of more accessible to a broader range of people. And notice this is something where if the researchers had just started with the 60,000 immunology papers, they would have been lacking information, kind of information that had been learned from training on these 1 million other scientific papers in other areas. At the same time, if they had started with a different corpus, maybe they had 1 million Reddit posts, that would probably be less useful because that is less similar to their goal of understanding immunology papers. So there's a balance here of wanting to find a larger data set, but that is still somewhat related to the data set you're interested in. In this case, they were able to extract 283,000 tuples of cell types, cytokines, and transcription factor relationships. And this was done by a tiny team of just 2 people, and that's part of the amazing power of transfer learning, is that it lets smaller teams do really interesting and impactful work. Previous work in this area, this idea of text mining immunology papers to gain new insights, has led to experimental validation of new immune cell cytokine relationships that were not previously documented. So this is a promising area. The third and final application of deep learning to immunology that I want to discuss is drug discovery. So here I'm going to talk about a paper in which deep learning was used to look for promising candidates for new antibiotics. And so there are many teams that are applying deep learning to antibiotic discovery, and it's also being applied to design or discover new therapeutic antibodies as well. So conventional small molecule screening is limited in how many molecules can be screened, and there tend to be very low hit validation rates for what is actually a promising lead. In contrast, using deep learning and computational methods can allow for searching a much larger chemical space of potential molecules. And then an iterative approach with machine learning can let teams zero in on the most promising molecules. In this case, the researchers were able to identify a new antibiotic, which they named halicin, and halicin was found to be effective against pan-resistant Acinetobacter baumannii. So Acinetobacter baumannii is rated by the WHO as one of the top 5 most urgent threats that we're facing with antimicrobial resistance in terms of species of bacteria that are now resistant to several types of antibiotics and for which we desperately need new classes of antibiotics for. Halicin was also found to be effective against C. diff and E. coli. The process that the researchers used, they started with kind of ranking the molecules by giving them prediction scores. And this is using a data set from the Drug Repurposing Hub, which is a really useful collection of molecules. And they rated over 4,000 molecules and used this prediction score to identify the most promising 99. From there, they tested them against E. coli and found that about half inhibited the growth of E. coli. Using this the smaller group, they then prioritized them based on looking at the clinical phase of investigation that they were under, their structural similarity to other molecules in the training data set, as well as the predicted toxicity in humans, because they want to find something that has low likelihood of being toxic to humans. And so here, this is showing halicin, which they identified as particularly promising, and its effectiveness against E. coli. Next, they tested it against pan-resistant Abamani. Again, this is an urgent threat bacteria that is resistant against many types of antibiotics. They showed that at different concentrations and different times, it was effective. And then they also tested it in a mouse wound. Here, showing that it inhibited growth of the bacteria. So I think this is really promising and an important area of direction. And as I mentioned before, so this is an example of antibiotic discovery, but deep learning is also being applied to the discovery and design of therapeutic antibodies. So whenever you're using any form of machine learning or AI, it's really important to think about the risk and gaps as well. There's a fantastic paper called "Everyone Wants to Do the Model Work, Not the Data Work" that interviewed dozens of machine learning practitioners and includes real postmortems on what can go wrong in practice when machine learning models are deployed in high-stakes situations. They identified kind of issues that arise in different, so here along the x-axis are the phases of creating and deploying a machine learning model, so starting with the problem statement through data collection and labeling, down to model selection and training, to deployment. And there are a number of things that can go wrong and which end up compounding and even cascading. One key one is interactions with physical world brittleness. The physical world is incredibly complex, and no model captures all that complexity. So there can be problems when the model is deployed and that complexity has not been adequately captured. Inadequate domain expertise of how this is being applied, missing data, conflicting reward systems. Cross-disciplinary work can be quite difficult when different disciplines have different practices and different incentives. Poor cross-organizational documentation, also a lack of mechanistic understanding of what's going on under the hood. So these are all risks to keep in mind and try to address before they surface in practice. Another very helpful paper on this topic is a framework for understanding sources of harm throughout the machine learning cycle. So there are many different sources of harm, again, that can come in at different points in the cycle of creating and deploying a model. So for example, databases of TCR protein structures are highly biased towards particular HLA alleles. Those are the genetics that determine what your MHC molecules can recognize. And so this is a form of representation bias, but a model, if it's not trained on certain HLA alleles, it's not going to give helpful insight about those alleles, and that can be a real oversight if you assume that everyone is represented when they're not. It's also important to recognize that representation does not solve all problems. There are other types of bias that can appear, such as measurement bias. So oxygen pulse meters are a tool to measure blood oxygen levels, and they are less accurate on people with darker skin. And so even if you include people with darker skin in your study, those readings will be less accurate, and this will impact the accuracy of the model and can have real world implications when deployed. And so it's really important to kind of understand the different phases and what can go wrong in these different phases. Another example I want to give is that T cell receptor data sets. I believe 96% of them just have data on the beta chain and not on the alpha chain. And so keeping that in mind, that's a kind of an example of missing data or representation bias, that they're lacking data on the alpha chain. So the flip side of some of these risks is that they do pave the way for or point to some opportunities. One is the opportunity to create new competitions with well-curated data sets. So we saw in the history section about the impact that competitions have had on advancing deep learning. In particular, we saw the power of ImageNet and the CASP protein structure prediction competitions in really spurring innovation and leading to the development of AlexNet for image recognition and AlphaFold for protein folding. Something I want to note about, so with this literature review that I conducted, I was really starting with, what data exist, where is deep learning already being applied. However, I think it would be even more important to do a project in the opposite direction and start with asking immunology experts, what are your most pressing questions? And then, is there data we could collect that would be helpful in solving those and in which deep learning could be usefully applied? I think too often the field is biased towards going in the direction which I went in, where we kind of see what data we have and go with that. And there's even been research on this of, people will sometimes change their research questions to fit the data that they have because they're lacking the data that they need for the questions they're really interested in. And so it's really important to be aware of this dynamic and to seek ways that we can kind of center immunology expertise and not just avoid questions because the data would be hard to collect or doesn't currently exist. It's super important to reward cross-disciplinary work. It's very important that teams have a deep understanding and respect for the underlying immunology, microbiology, and deep learning, and aren't just overemphasizing one area. And then I wanted to share a really promising example from NeurIPS, which is a prestigious AI conference. The organizers realized several years ago that work on mathematical models was being kind of disproportionately incentivized, whereas there were very few incentives for kind of data sets and benchmarks, which are also quite important. And so they introduced a new track to the conference on equal standing with the original track. And I thought that was a great approach and offers a nice model of how we can further incentivize data set and benchmark work. Here are the references that I cited in this presentation, and then there are far more in my literature review on this topic. I encourage you to check out these papers. I think this is a really, really exciting area to be reading about. I love talking about immunology and deep learning, so please reach out to me if you're interested in these topics as well, because I would love to discuss them. Thank you.